recording. I probably should have recorded all that I was talking about. I graded lab 5, so it's now graded. And you can take a look at it and resubmit it by 11:59 p.m. this Saturday. I'll try and get labs 4 and 5 graded this weekend, as well as quiz 2A. Let's see, let's share my screen. There we go. I got an email here I got to read. There we go, the syllabus. We're going to be starting chapter 5 today. I'm pretty sure I finished chapter 4. We did talk about the endosymbiotic theory and discuss the proof of the theory of why eukaryotic cells have mitochondria and chloroplasts in them, and why these organelles have a DNA molecule in them, as well as ribosomes, which are 70s in size. Any question about any of that? If not, I'll begin chapter 5. In the lab, we do have a lab today. It's going to be lab 6. It's diffusion and antibiotics. So there will be a lab this Thursday. I'll just minimize that and go to chapter 5. Chapter 5, microbial metabolism. As usual, I'll give you an introductory slide, which lists the major goals and a rough, brief outline. So know the terms: catabolism, anabolism, activation, energy. What's an enzyme? Active site of an enzyme. Competitive inhibition, non-competitive or allosteric inhibition. What's a redox reaction? What are photoautotrophs, photoheterotrophs, chemoautotrophs, chemoheterotrophs? And remember, if you want to know all of the goals, these are just the major goals. Take a look at the objectives, which will be in the study guide for chapter 5. So take a look at the study guide, and the very start of that, there will be objectives. And if you know all of the objectives, you should do very well on the quizzes, at least for the lecture part, because the objectives are, well, essentially everything we'll cover. There we go, chapter 5, microbial metabolism and the objectives. And let's see how many there are. There's 19 objectives. While I'm here, let me open up lecture 4, chapter 4. Let me see if there's a regular chapter 4. There it is. All right, so chapter 4 is the notes. The lecture slides are now available online. If I finish a chapter and I don't do that, you guys can remind me. I'm trying to check to make sure I got chapter 3 up. Yeah, it's up. All right. So let's begin. Besides knowing the terms, know what feedback inhibition is, and then know what aerobic respiration is, its steps, and how it differs from anaerobic respiration. We will talk a large amount of this lesson about aerobic respiration, and then we will briefly compare it to anaerobic respiration. Any question about what we're doing? If not, let's begin. Microbial metabolism discusses obviously metabolism, which is all of the chemical reactions occurring in an organism. And since we're mainly talking about bacteria, you can say it's all of the chemical reactions occurring within a cell. because one cell makes up all of the microbes, at least all of the species. Even the microbes which, well, I shouldn't say all of the microbes, all of the bacteria, even the bacteria which are colonial, we consider them single celled because if you break one cell off the colony, the cell will continue growing, forming another colony. So we say that they are single cells as opposed to a multicellular organism. Any question about any of that? We can break metabolism down into catabolism and anabolism. Catabolism is the decomposition reactions where you start with a larger molecule, complex molecules such as starch, and you break it in a catabolic reaction to make smaller molecules. If it's starch, it breaks into glucose. When you do that, of course, energy is released and some of that energy is converted to heat. Whenever you convert one form of energy or one form of whatever, one form of energy to another, some energy is always converted into heat, and then we say it's lost because heat is not used by much. We'll just word it that way. It's almost not used by anything, but warm-blooded organisms can use that heat to help warm the organism. Okay. Catabolic reaction can transfer energy from the complex molecules, making the smaller molecules, into ATP. But this slide is a little bit oversimplified when it's showing complex molecules breaking down to simple molecules, making ATP, because in our cells, this only happens by two catabolic processes, and that is by aerobic respiration, making ATP, and by fermentation, making, or I should say glycolysis and fermentation, or just glycolysis making ATP. Okay, our cells are very lousy at converting one form of energy into making ATP and then converting that larger molecule into smaller molecules, meaning all of the catabolic reactions occurring in your cells, with the exception of two, do not make ATP. Is everyone clear on that? The majority of catabolic reactions, the vast majority of catabolic reactions in your cells do not make ATP. Even in green plants, which are much better, much more efficient at making ATP, meaning they have more reactions, more catabolic reactions to make ATP than our cells have. They're not very good at it. Only a few catabolic reactions can make ATP. The majority of the catabolic reactions do not make ATP, such as when we break down sucrose into glucose and fructose. That is a catabolic reaction, but our cells cannot make ATP out of it, and green plants cannot either, and bacteria can't either. Any question about any of that? All right, an anabolic reaction is a biosynthesis reaction, and it consumes energy, meaning energy has to come into the reaction in order for it to happen. And an anabolic reaction, the simple molecules, such as glucose, are converted into a larger, more complex molecule, such as starch. Whenever you convert energy from one form, like glucose, into starch, another form of energy, some energy is always lost as heat. Any question about that? So as I mentioned, whenever energy is converted from one form or another, some of it is always converted into heat. Anabolic reactions require energy in order for them to occur. Otherwise, you cannot get the simple molecules forming the larger molecules. because a larger molecule has more energy in it than the simple molecules. They take up energy. That energy usually comes from ATP, breaking down into ADP and phosphate, releasing the energy needed by the anabolic reaction to occur. All right, any question about any of that? You can link metabolic reactions together so that molecule A can be converted into molecule B, converted into molecule C, converted into molecule D, converted into molecule E, converted into molecule F. When you link them together, we call that a metabolic pathway. It's a sequence of enzymatically catalyzed chemical reactions in a cell. Blow this up. In a cell, all of these chemical reactions will have an enzyme which will catalyze the conversion of one molecule to another molecule. The first molecule is called the starting substrate or the reactant, and then the end molecule is called the end product. All of the in-between molecules are called intermediate products because molecule B is a product of this reaction, molecule C is a product of this reaction. So these are intermediates or intermediate products. As I stated, in cells, we have an enzyme to convert one molecule into another. So metabolic pathways are determined in cells by enzymes, and enzymes are coded by genes. So it's all in your genetics. Any question about any of that? All right. Cells will have metabolic pathways to convert one molecule into an end product. Here are all the chemical reactions occurring within a typical cell. You only have to know all of them, okay? Any questions? Just kidding. You don't need to know all of those, but it's telling you that there's a lot of chemical reactions occurring within a typical cell. I think that right there is aerobic respiration, or at least the Krebs cycle of aerobic respiration. I'd have to blow it up to be certain, but it looks like it. Chemical reactions occur according to the collision theory. The collision theory states the chemical reactions occur when atoms, ions, or molecules simply collide with each other and they have enough energy to form the product. Obviously, if it's very cold and they collide, they won't have enough energy when they collide to form the product. But if they have enough energy, they will form the product. The activation energy is the energy needed to disrupt the electronic configurations in the starting atoms, ions, or molecules. And then when they're disrupted, they make new electronic configurations, which will be when the product is formed. The reaction rate is the frequency of collisions with enough energy to bring about a reaction. You can increase the reaction rate by A, increasing the abundance of enzymes. The more enzymes there are, the faster they can convert the reactants to the products. Or B, by increasing the temperature or the pressure. I think you understand if you increase the temperature, what you're doing is you're making the molecules move faster. And if they're moving faster, they have more energy. And if they have more energy, when they collide, they'll be more likely to have the energy they need to make the product. meaning if they have more energy they have, the more likely they'll have the activation energy they need to make the product. So I think you understand increasing the temperature. Increasing the pressure does the same thing. When you increase the pressure, first of all, when you do that, you bring the molecules closer together so they're more likely to collide. And when you increase the pressure and bring the molecules closer together, they have more energy in it so that when they're more likely to collide, they're also more likely to have more energy. And so you can increase the reaction rate. And you don't really need to understand pressure that well, but just understand that pressure also increases the reaction rate. Any question about any of that? So enzymes reduce the activation energy of a chemical reaction. Here we're seeing a chemical reaction, and on the y-axis we have the amount of energy in the system. On the y-axis, the x-axis is just time. So initially we have two molecules that we're going to call reactants, and they have a given amount of energy with them. And actually, I guess that's just one molecule linked together, A and B linked together. And they have more energy in them, the reactants, than they have the products. And this could be something like sucrose has more energy in it than glucose and fructose. And so if I had a beaker of sucrose and then just left it in the beaker, over time, the sucrose would convert into glucose and fructose. Okay, any question about that? There should be a question. Nobody's going to ask? If you have a beaker of sucrose, how quickly does it turn into glucose and fructose? Come on, that's the time frame for an answer. Just give me a time. Does it happen within a second, a minute, an hour, a day, a month, a year? All right, well, think about that. How often does that happen? Obviously, you've seen a beaker of sucrose, and generally speaking, it doesn't usually turn into glucose and fructose, but it will with time, because the molecules will collide, and every once in a while, they will collide with enough energy to have enough energy to perform the chemical reaction. The trouble is that without an enzyme around, you need a lot of energy before the chemical reaction can occur. And that we call the activation energy without an enzyme. So this molecule has to get way up here, and that we call the energy hump, before it can come down here. And I forgot to mention, the products have less energy than the reactant in this case. And that would be the case if we're starting with sucrose. It has more energy in it than fructose or glucose, even when you put those two together. Any question about that? And that energy hump is so high that sucrose does not often break down into fructose and glucose. And that's what I'm asking you, how long does that take? Nobody's going to comment. All right. And yet, sucrose in your cells breaks down into glucose and fructose all the time. And why is that? Hello? You have to participate. Boy, you guys
Will you
really don't.
repeat the question?
In your cells, sucrose breaks down into glucose and fructose all the time. And why is that?
Oh, I don't know.
For energy?
No, your cells don't have more energy because the energy you need to, well, that's not true, or the energy you need to break down sucrose and fructose. The energy you need to break down glucose into carbon dioxide and water, what you would need to do is take a blowtorch to the glucose. But if you did take a blowtorch to it, you would get the glucose up to a high enough energy that the activation energy would be reached, and you convert that glucose into carbon dioxide and water. I don't know how much energy you need to break sucrose down into fructose and glucose, and so that's why I changed the question, because I don't know how much energy you need to break down sucrose. I just don't know. Okay, it's a lot of energy, but it's probably not as high as a blowtorch. And if you don't know, a blowtorch is something like 1,000 degrees. I think that's 1,000 degrees Fahrenheit. And even if it's not 1,000 degrees, like maybe it's only 800 degrees Fahrenheit, it's still really, really hot. Okay. It might be over 1,000. I don't know what the temperature of a blowtorch is, but it's really, really hot. All right. And that is not the energy you have in your cells, because if your cell were ever to go anywhere near 1,000 degrees Fahrenheit, that cell would instantly die and be turned into charcoal. Okay. The reason why this reaction happens in your cells all the time is because your cells have an enzyme. And the activation energy with an enzyme is much less than the activation energy without an enzyme. And this scale is not two scale, meaning the amount of energy you need in your cells for this to happen is only about 37 degrees, your body temperature. Okay. And then, as I stated, converting glucose into CO2 and water, you need to get that someplace near 1,000 degrees Fahrenheit, not 98.6 degrees Fahrenheit. Okay. So that's why this reaction is happening in your cells all the time, because your cells have enzymes which can perform this reaction. And with the enzyme, this reaction is much more likely to happen because the activation energy with the enzyme is much, much less than the activation energy without the enzyme. Any question about any of that? This is a very important graph, so make sure you understand it. We will be talking about that well, not all today, but a lot of today when we're talking about aerobic respiration. All of those reactions of aerobic respiration happen because of this, at least if they're a catabolic reaction. Now this is showing you a catabolic reaction, and you know that because the end product has less energy than the reactant. So biological catalysts speed up chemical reactions. And in cells, we have biological catalysts, which we call enzymes. And they can speed up a chemical reaction 10 to the 8th or 10 to the 10 fold times. If you remember, 10 to the 6 is 1,000,000 times. So this is, I don't know, within a magnitude of 1,000,000,000 times. Did I say that right? Yeah. Meaning 10 to the 9th is 1,000,000,000 times. And biological catalysts are enzymes, and enzymes are encoded by genes. enzymes are specific for performing one single chemical reaction. The specificity is due to the enzyme's three-dimensional shape. And I'll talk a little bit more about that. For this class, we're going to talk about enzymes a little bit oversimplified. You might know from working in the clinic or something like that, but we can have a drug that mimics the substrate, and then that fouls up the enzyme because the enzyme will bind to or react with the substrate, which is the drug, instead of the real substrate, meaning there are some enzymes which can bind to something else, but they're very specific. And for a little bit of an oversimplification, we're going to say an enzyme essentially binds to only one thing, because even if they bind to two things, it's still very specific because there's lots of molecules in our cells, and an enzyme isn't going to bind to almost all of them. So for making this class a little bit simpler, let's say that an enzyme will only bind with one substrate. And I'm already telling you that's a little bit oversimplified. We can find a drug, for example, which can then bind to the enzyme as well. But other than that, enzymes are very specific. And they're very specific because of their three-dimensional shape. So an enzyme has a specific shape, and it will only bind to one specific substrate. Enzymes are also not used up in a chemical reaction. So when an enzyme performs one chemical reaction, when it's done, that same enzyme can bind to another substrate and perform another chemical reaction. And that can happen many, many times. That's why catalysts can speed up a chemical reaction 10 to the 8th to 10 to the 10th times. Enzymes also bind to the substrate, so they form an enzyme substrate complex. So when the substrate comes to the enzyme, the two actually bind, and then that orients the substrate, increasing the probability that the reaction will occur. It puts stress on the chemical reactions of the substrate, and then makes it more likely that the chemical reactions of the reactant will occur. And that lowers the activation energy. Is that confusing? I've got another picture, which I'll discuss that in a little bit. And I'll discuss this a little bit more too, the specificity. You should know that enzyme names typically or normally end with ACE. We've already talked about catalase. We will talk in this lesson about ligase. So words that end in ACE are typically an enzyme. Not all enzymes end in ACE, like renin is an older enzyme, and I think it was named before the ACE convention, but there are a few enzymes which don't end in ACE, but most of them do end in ACE. You do not need to know the examples. I'm just showing you some different examples of enzymes. You should know that enzymes do not work in a vacuum, that some enzymes require either a coenzyme or a cofactor, which helps out the enzyme. And then when the coenzyme or cofactor binds to the enzyme, it makes the complete enzyme so that the complete enzyme can now bind to the substrate. You do not need to know the name apoenzyme or holoenzyme, but you should know a coenzyme or a cofactor. A coenzyme is a biological molecule which helps out an enzyme. And we'll talk about a few coenzymes in this lesson, sorry, like NAD plus is a coenzyme. We'll talk about that in aerobic respiration. The difference between a coenzyme is a little bigger. It's a biological molecule. And a cofactor is usually a non-protein activator, such as an element, usually a metal like iron, zinc, magnesium, calcium. So that's how a cofactor differs from a coenzyme. Any question about that? Some important coenzymes are NAD, NAD+, and you can know it by that name, or by nicotinamide adenine dinucleotide. I'm sure most of you will call it NAD+. We will talk about this one because you run into it in aerobic respiration. FAD, or flavin adenine dinucleotide, is also one we'll talk about because we'll run into it in aerobic respiration. The other coenzymes we're not going to talk about because we don't run into these two in aerobic respiration. I probably should require you to know coenzyme A because we do run into it in aerobic respiration, but I'm not going to require it. I never have. Just know the first two, and we're going to talk about them in pretty good detail when we talk about aerobic respiration a little bit later on in this lesson, and that might be next Tuesday. I'm not sure how far we'll get in aerobic respiration today. So an enzyme is very specific to binding to a substrate, and an enzyme has an active site. That is the region of the enzyme that binds to the substrate. And the active site has a three-dimensional shape, and the substrate will bind in that active site. And that binding is so specific, that's the word I'm looking for, that we can say an enzyme fits a substrate sort of like a lock and key fit each other. Meaning, generally speaking, there's one key that can open a lock. And of course, there's a master key. So there's the regular key and then the master key. So there are a couple of keys that can open the lock, but you're getting the idea. And we can say that an enzyme follows the lock and key model because one enzyme is specific for one substrate. And that one enzyme can only bind specifically to one substrate. And when they bind, they change the electronic configuration of the bonding in the molecule of the substrate, and that puts stress on those bonds, and that will help create the product, which has new energy bonds in it. Okay, so this is showing you that the binding of the enzyme to the substrate puts stresses on the bonds of the substrate, and that helps convert the substrate into the product. The product then falls off of the enzyme, and then that enzyme can re-bind to another substrate and perform another chemical reaction. So that's the lock and key model of an enzyme. Any question about that? All right, I'm going to ask you a few questions. Here is another model of an enzyme, and this is closer to a real one. And here's the substrate, and there's the enzyme. And this is all of those dots are amino acids in a protein. So there's a lot of them. And I guess all of those would be either molecules in the substrate or atoms in the substrate. And then the substrate will bind to the enzyme in the active site, which is right here. And there we can see the enzyme substrate complex, forming a complex where they're bound together. And what do you notice about this picture? There's three things you should recognize. What's the first thing you can recognize? Very easy, the first one.
I guess it kind of looks like the enzyme is swallowing the substrate.
Well, yeah, they're binding to each other, if that's what you mean. What else do you notice about the substrate and the enzyme?
The shape, or
What about the shape?
one's large and one's small.
Yeah, the enzyme is much larger than the substrate. That was the first thing you're supposed to learn from this. The enzyme is very many times larger than the substrate. Okay, what's the other easy thing to recognize here? There's two things that are easy to see here.
The shape of the enzyme fits directly into the shape of the substrate, like the lock and key thing that we were talking about earlier.
Yeah, we've already discussed that in the lock and the key. So I'm looking for something different that is not in the lock and key model. Let's look here and then look here. What's the difference? The enzyme. What's different about the enzyme here and here? Come on, somebody's got to see that. It's pretty obvious.
Say it again.
What's different of the enzyme here and here?
One's in, one's out.
Yeah, what else is different?
One's open, one's closed.
I think I'll accept that. And that is this enzyme has changed its shape. So this is not the lock and the key model because the lock and the key don't change. This enzyme changes its shape when it binds to the substrate. And this we can call the hand in the glove model, where the glove molds to fit the substrate. The enzyme changes its shape. Okay? The third thing is very difficult to see. Maybe I can blow this up and you guys will see it better. And that is if you look real closely at the substrate here and the substrate there, this substrate is a little flatter than this substrate, meaning the substrate has changed its shape when it binds to the enzyme. And I like to say that the shapeshifter's hand molded to fit the glove as the glove was molding to fit the hand. And those of you who know Star Trek Deep Space Nine will know what I'm talking about when I say a shapeshifter's hand is molding to fit the glove, which is molding to fit the hand. And that's beyond the hand in the glove model. It's the shapeshifter's hand molding to fit the glove, as well as the glove molding to fit the hand. Any question about that? So that's what's happening with the enzymes, okay? And that's a lot going on in this slide. There are a number of factors which influence the activity of an enzyme: the temperature, the pH, the salinity, the presence of heavy metals, alcohol, UV radiation can all influence the activation or the activity of an enzyme. These can all cause protein denaturation. Denaturation is where the enzyme loses its shape. And as it loses its shape, it will perform less and less well until it totally becomes denatured, and then the enzyme will not have any activity. The substrate concentration can also influence how much activity the enzyme has. Generally, as you increase the amount of substrate, the enzyme will have more activity, but that only happens up to a point. We'll talk a little more about that. There are poisons which can affect the enzyme activity, and you can see a cyanide or fluoride can stop the enzyme from acting. And some of these poisons totally denature the enzyme, and then the enzyme can't act. You can have inhibition, such as competitive inhibition, stopping the enzyme from having activity. And then there's also non-competitive or allosteric inhibition. We'll talk about both of these in just a little bit. And then there's also feedback inhibition. All of these factors can influence how much activity an enzyme has. Let's talk about each of these in a little more detail. I mentioned that an enzyme can be denatured. So this is the folding pattern of a functional enzyme, and it has a specific three-dimensional shape. And you'll remember there will be binding, like hydrogen bonding between here, this amino acid here and that amino acid there. There can be ionic attraction, like between this one and that one. There can be repulsion, like maybe right here and right there. And in the wrong environment, such as the wrong salinity, the wrong pH, or the wrong temperature, the protein can denature and unfold. And this one's totally unfolded, it's denatured, totally denatured, and then the enzyme will have no activity. Now, you can have it partially unfolded, in which case the enzyme has lost partial activity. Why did the protein, this protein, become denatured at the wrong temperature, the wrong pH, or the wrong whatever? Why does the protein unfold at the wrong temperature or the wrong pH? Nobody's going to guess? It's because at the wrong temperature, the wrong pH, the binding pattern gets disrupted. Like if there's a hydrogen bond right here and right there, hydrogen bonds are weak, and at the wrong temperature, that bond can break, and then this portion will unfold. Okay, and that's true for the wrong pH or the wrong other environment too. And when it's way off, you can break the ionic bonds. What other one? We'll just leave it there. All right, any questions? This graph is showing you that enzymes typically work best and have an optimal temperature that they work at. This enzyme is an enzyme that works best at around 37 degrees. So this is an enzyme that works best in our body temperature. And that could be most of the enzyme in your cells, as well as an enzyme in a human pathogen. Its enzyme will work best at body temperature. And as you move away from body temperature, the enzyme will work, but the further you move away from 37 degrees, the less activity the enzyme will have until you move at an extreme, in which case the enzyme is totally denatured. It will no longer work. And that's true of the pH. And this one here, it has the optimal pH around pH 5, and the enzyme has the most activity around pH 5. And as you move away from pH 5, the enzyme has less activity. And then at extreme pHs, the enzyme has no activity. It's been totally denatured. What enzyme in your body will work best around pH 5? Nobody knows? Nobody wants to guess? It's not your stomach, because that goes down to pH 2 or even pH 1. So that enzyme would work best around pH 2. So what enzyme works best around pH 5? We'll think about that. And if you get the answer, go ahead and just state it. Enzymes have more activity when you increase the substrate. So when we increase the substrate over here, we note that the enzyme activity increases up to a point. When we get here, when we increase the substrate concentration, the enzyme activity no longer increases. Why in this portion did the enzyme activity increase? And then right here, when we add more substrate, the enzyme is not increasing its activity. Okay, nobody wants to guess. We'll move on. Competitive inhibition. Factors influencing enzyme activity. A competitive inhibition is an inhibitor that competes with an enzyme. It competes actively with the active site. And so we can have a drug which acts as a competitive inhibitor. And then that drug will bind to the enzyme in the active site, and that will stop the substrate from binding to the active site of the enzyme. And that's competitive inhibition, where the inhibitor binds in the active site of the enzyme. Any question about any of that? All right, no questions. Let's move on to non-competitive inhibition, which is also called allosteric inhibition. This is when the inhibitor interacts with the enzyme at a site other than the active site. So here's the active site, here's the substrate which binds to it. The allosteric inhibitor binds at a site other than the active site. And the allosteric inhibitor, when it binds to the enzyme, it changes the shape of the enzyme, changes the folding pattern of the enzyme. And that changes the shape of the active site. And now the active site can no longer bind to the substrate, and that will shut down the enzyme. So this is allosteric inhibition, meaning the inhibitor bound at an allosteric site. And it's also called non-competitive inhibition. Any question about any of that? Feedback inhibition is where we have an enzymatic pathway. Let me blow this up. So we have one molecule, substrate, which is converted to intermediate A by enzyme 1. And then intermediate A is converted to intermediate B by enzyme 2. And then intermediate B is converted into the substrate by enzyme 3. So we have an enzymatic pathway here. And you can have feedback inhibition where the end product, when it builds up in the cell, can then bind to the enzyme, one of the enzymes, usually the first enzyme, and then act as an allosteric inhibitor, changing the binding pattern of the enzyme so it can no longer bind to the substrate. And this is feedback inhibition, where the end product itself, when it builds up in the cell, will shut down the enzymatic pathway. You don't have enzyme 1 functioning, so you can't get intermediate A. If you can't get intermediate A, you can't get intermediate B. If you can't get intermediate B, you can't make more end product. Why cells usually have a feedback inhibition this way, where the end product, when there's enough of it in the cell, will bind to the first enzyme and then shut the first enzyme down, is that if the cell were to shut down enzyme 2 or enzyme 3, it will be less energy efficient. Meaning if we shut down enzyme 2, the cell will make intermediate A when it doesn't need it because the cell has enough end product. Okay, so usually the cell will have the end product bind to the first enzyme and then shut it down. And that way, none of the substrate will be converted to any of the intermediates or to the end product. And then why cells often have this feedback inhibition is that when the end product decreases in concentration in the cell, and the cell needs more end product, there won't be enough end product to bind to the enzyme. And so enzyme 1 will be active and will make intermediate A, and then intermediate A will make intermediate B, which will then make the end product. Now, I haven't stated it, but ever so often, if we have an enzyme which is shut down by the end product, this binding of the end product to the enzyme 1 happens by a stoichiomic reaction, and every once in a while, the end product will fall off. And so enzyme 1 will then be active. But if you have enough end product around, one of them will bind to the enzyme and inactivate it. So we don't have this pathway. But if there isn't enough end product in the cell, there won't be enough of it to bind to the enzyme. And then the enzymatic pathway will proceed until more of the end product gets made. So that's why cells frequently have a feedback inhibition loop like this in the cell, where the enzyme, where the end product actually shuts down the first enzyme in the enzymatic reaction. Any question about any of that? All right, if there's no questions, let's move on to ribozymes. Ribozymes are non-protein enzymes. They are enzymes, but they're not made up of proteins. Ribozymes are RNA enzymes. They are RNA that cuts and splices RNA or another molecule, and they function as enzymes in that the RNA has an active site that actually binds to the substrate, and then the RNA is not used up or modified in the chemical reaction. So once it performs the chemical reaction, this ribozyme can then bind to another substrate and perform another chemical reaction. So ribozymes are enzymes, okay? They're just special because most enzymes are made up of protein. Probably over 95% of enzymes are protein enzymes, but there are some which are RNA enzymes, and you need to know that. Okay, there's only one RNA enzyme we'll ever discuss in this class, and that is in the ribosome. The molecule which takes the amino acid that is brought by tRNA to the growing polypeptide chain to make an enzyme is linked together by the ribosome. And the portion of the ribosome that does that is an RNA portion of the ribosome. So this is the only enzyme that we will discuss, which is a ribozyme, meaning an RNA enzyme. Any question about any of that? All right. Let's move on. There is metabolic diversity among all organisms. All organisms, including microbes, require the following: an energy source and a carbon source. We can classify organisms by their energy source. If an organism gets its energy from light, we classify those organisms as a phototroph. Phototroph means they derive their energy from light. And obviously, green plants and green photosynthetic bacteria are phototrophs. If, on the other hand, the organism gets its energy needs from a chemical, we call them a chemotroph, and they derive their energy from an inorganic or organic chemical compound. We are chemotrophs, and we get our energy needs from the food we eat or drink, largely glucose, but it can be any food molecule. Many bacteria do the same, and we call them chemotrophs, like E. coli, and they get their energy needs from an organic chemical compound, such as glucose. But there are some prokaryotes which can get their energy needs from an inorganic chemical compound. And these bacteria or archaea are chemosynthetic organisms. And we'll talk a little bit more about them. But there are some organisms which can get their energy needs from an inorganic chemical compound. I'm talking about a molecule like ammonia or iron or sulfate. You can get their energy needs from those molecules. Any question about that? If, on the other hand, an organism gets its carbon source, we can classify them by where they get their carbon source. They are an autotroph if they can get their carbon source from CO2, either in the air or dissolved in the water. So an autotroph gets their carbon from CO2. On the other hand, if they get their carbon from an organic chemical compound, such as glucose, we classify them as a heterotroph. And we are a heterotroph. We get our carbon source from the food we eat, largely from glucose. E. coli is another example of a heterotroph. It gets its carbon from the food molecules it eats, which is largely glucose, but it could be another food as well. Any question about that? Now, if you ever have confusion on what something is, this is the easiest way to break it down. Say, what's its energy source? If it gets its energy from light, call it a phototroph. If it gets its energy needs from a chemical, call it a chemotroph. And then break it down its carbon source. If it gets its carbon from CO2, it's an autotroph. And if it gets its carbon from an organic chemical compound, it's a heterotroph. Because we can now combine energy source and carbon source, and then we say that the organism can be classified into one of four types. There's the photoautotrophs, and they get their energy needs from light, and their carbon from CO2. Those are photoheterotrophs. They get their energy needs from light, and they get their carbon from a chemical organic compound like glucose. There's the chemoautotrophs, and they get their energy needs from a chemical compound, and they get their carbon from carbon dioxide. And then there's the chemoheterotrophs. They get their energy needs from a chemical compound, such as glucose, and they get their energy needs, let's see, I already said that. They get their carbon needs from a organic compound, such as glucose. Any question about any of that? All right, we can talk about the 4 different trophs right here. And the photoautotrophs get their energy need from light and their carbon source from CO2. You can have 2 types of photoautotrophs, those which are oxygenic, and green plants are oxygenic, meaning they engage in photosynthesis and release oxygen. But you can also have anoxygenic bacteria, and they do not release oxygen. You're probably not familiar with an anoxygenic photoautotroph, but the green and purple sulfur bacteria are anoxygenic photoautotrophs. Any question about that? There are photoautotrophs that you probably are familiar with, which are bacteria. Cyanobacteria is a photosynthetic bacteria, and it is oxygenic photoautotroph. We're going to skip the photoheterotrophs and talk about it last because you're probably not familiar with them. And we'll talk about the chemoheterotrophs next because they are something that you're familiar with. Chemoheterotrophs get their energy needs from a chemical compound, and this can be an organic chemical compound like glucose, and that would be us. We're a chemoheterotroph, getting our energy needs from glucose or some other food. But there can be a chemoheterotroph which gets its energy needs from an inorganic chemical compound, such as iron or ammonia or sulfate. There are some chemosynthetic bacteria which can utilize a different molecule and get their energy needs from a chemical compound, and it's usually an inorganic chemical compound. The chemoheterotrophs get their carbon source from an organic chemical compound such as glucose, and that's where we get our carbon source. You'll note that we get our carbon source and our energy source generally from the same molecule. Like if you're eating a bunch of sugar, you'll get your energy needs and your carbon needs from glucose. But we get it from some food molecule, both of these. And with many chemoheterotrophs, they can get the energy source and the carbon source from the same molecule. It doesn't need to be the case, especially with the chemosynthetic bacteria. They can use one molecule to get their energy needs, and that could be maybe iron, for example, to get their energy needs. But then they will get their carbon source from an organic chemical compound, such as glucose. And I mentioned that we're that way, all animals are that way. The fermentative bacteria are that way. Some bacteria like E. coli are this way. And then some of the chemosynthetic bacteria are this way. Now the chemoautotrophs are chemosynthetic bacteria, which get their energy needs from an inorganic chemical compound, and they get their carbon needs from CO2. So you'll notice that the chemosynthetic bacteria, some of them are chemoautotrophs and some are chemoheterotrophs. Okay, you actually know a chemoautotroph, you just don't realize you know it. If you lift up the back of the water tank of the toilet, I'm not talking about the toilet bowl, I'm talking about the toilet tank, you'll see yellow or brownish scum on that. That's iron oxidizing bacteria utilizing iron in the water to get its energy needs. And then it gets its carbon source from CO2. So there's a chemoautotroph that you're familiar with. The photoheterotrophs get their energy needs from light, and they get their carbon source from an organic chemical compound, such as glucose. You're probably not familiar with the photoheterotroph, but the green and purple non-sulfur bacteria are photoheterotrophs. All right, any question about any of this? If there's no questions, I'm going to stop here and we'll continue this lesson next time. And I'll see you at 6:30 for the lab.